Hello students and welcome to the lecture on Functional Anatomy of Respiratory System, Mechanism of Pulmonary Ventilation, Alveolar Ventilation, Gaseous Exchange, Transport of Gases in Blood. Let us begin with the definition of the respiration. Respiration is the exchange of oxygen and carbon dioxide between environment and cells of the body where organic nutrients are broken down enzymatically to release energy. The process of gas exchange in the body, called respiration, has three basic steps. Number 1, pulmonary ventilation or breathing is the inhalation and exhalation of air between the atmosphere and the alveoli of the lungs. Air flows between the atmosphere and the alveoli of the lungs because of alternating pressure differences created by the contraction and relaxation of the respiratory muscles, which will be discussed later. Number 2, external respiration is the exchange of gases between the alveoli of the lungs and the blood in the pulmonary capillaries across the respiratory membrane. In this process, pulmonary capillary blood gains oxygen and loses carbon dioxide. Number 3, internal respiration is the exchange of gases between blood in systemic capillaries and tissue cells. In this step, blood loses oxygen and gains carbon dioxide. Within cells, the metabolic reactions that consume oxygen and give off carbon dioxide during the production of ATP are termed cellular respiration. Now, let us start with the first part of our topic, that is functional anatomy of respiratory system. The respiratory system consists of upper and lower respiratory tracts. The upper respiratory tract includes the mouth, nose, nasal cavity, pharynx, and larynx or voice box. Each has a specific function to aid the flow of air into the body. Let us briefly discuss them one by one. Upper respiratory tract, mouth, nose, and nasal cavity. The function of this part of the system is to warm, filter, and moisten the incoming air. Pharynx, here the throat divides into the trachea and esophagus, that is the food pipe. There is also a small flap of cartilage called the epiglottis, which prevents food from entering the trachea. Larynx, it helps to protect the trachea by producing a strong cuff relax if any object passes the epiglottis. Parts of the lower respiratory tract. Trachea, also known as the windpipe. This is the tube which carries air from the throat into the lungs. The inner membrane of the trachea is covered in tiny hairs called cilia, which catch particles of dust which we can then remove through cuffing. The trachea is surrounded by 15 to 20 C-shaped rings of cartilage at the front and side which help protect the trachea and keep it open. Bronchi. The trachea divides into two tubes called bronchi, one entering the left and one entering the right lung. Once inside the lung, the bronchi split several ways, forming tertiary bronchi. Bronchioles. Tertiary bronchi continue to divide and become bronchioles, very narrow tubes less than 1 millimeter in diameter, and leads to the alveolar sacs. Alveoli. Individual hollow cavities contained within the alveolar sacs. Alveoli has very thin walls which permit the exchange of gases, oxygen and carbon dioxide. They are surrounded by a network of capillaries into which the inspired gases pass. Diaphragm. The diaphragm is a broad band of muscle which sits underneath the lungs, attaching to the lower ribs, sternum and the lumbar spine and forming the base of the thoracic cavity. Dear students, I would like to briefly discuss the lungs. The lungs occupy all the space of the thoracic cavity except for the mediastinum. The lungs are located in two chambers of the thoracic cavity on either side of the heart. Though similar in appearance, the two lungs are not identical nor wholly symmetrical. Fissures of double folds of pleura divide the lungs into lobes. The lobes are further divided into two segments and then into lobules. Each lobe contains a number of bronchopulmonary segments, each served by its own artery, vein, and tertiary bronchus. There are two circulations that serve the lungs. The pulmonary network carries systemic blood to the lungs for oxygenation, and the bronchial arteries provide systemic blood to the lung tissue. The lungs are innervated by parasympathetic and sympathetic motor fibers that constrict or dilate the airways, as well as the visceral sensory fibers. Moving to the second part of today's lecture, that is the mechanism of pulmonary ventilation. The morphological components of the respiratory system are the airways and the lung parenchyma consisting of pulmonary acini. The respiratory airways ensure the transport of gases and improve the quality of the inspired air, while the pulmonary acini are involved in gaseous exchange. The pulmonary acinus represents the morphofunctional unit of the lung. Pulmonary ventilation is comprised of all mechanical processes that ensure the exchange of gases between the atmosphere and the lungs. Through ventilation, oxygen-rich air is introduced into the pulmonary and alveoli during inspiration, and carbon dioxide-rich air is eliminated from the lungs into the atmosphere during expiration. Gaseous exchange between the atmosphere and the lung take place due to differences in pressure gradient between the two environments. These differences are due to the variations in lung volume, which occur because the lungs follow passively the movements of the thoracic wall. The resulting pressure gradient will cause a movement of air from high pressure environment to low pressure environment. That is explained by Boyle's law. A respiration rate higher than the normal limit is called tachypnea, and the low rate is called the bradypnea. Now, moving on to the role of muscles in inspiration. Lungs do not possess muscular elements and cannot initiate on their own volume changes characteristic to respiratory phases. Therefore, they will follow passively the movements of the chest, to which they are connected through the pleural system comprising of parietal pleura and the visceral pleura. The lungs can be expanded and contracted in two ways: by downward and upward movement of the diaphragm to lengthen or shorten the chest cavity, and by elevation and depression of the ribs to increase and decrease the anterior-posterior diameter of the chest cavity. Further, the pressure changes that occur during the process of ventilation and their effect on the mechanism of ventilation will be discussed as: number 1, pressure changes during inspiration. During inspiration, atmospheric air enters the lungs because the pressure of atmospheric gas is higher than the intrapulmonary or intra-alveolar pressure. Inspiration is triggered by stimulus generated in the inspiratory center of the medulla oblongata, which travel to the motor neurons of the anterior spinal horns. Through the spinal nerves, the impulses contract the inspiratory muscles. During resting inspiration, the diaphragm and the external intercostal muscles contract. By contraction, the diaphragm flattens and lowers by approximately 1.5 to 2 cm during resting inspiration and 7 to 8 cm in forced inspiration. By lowering, the diaphragm increases the longitudinal diameter of the thoracic cavity. The volume increase resulted by contracting these muscles allow a quantity of air to be introduced into the lungs. The contraction of the external intercostal muscles cause the ribs to horizontalize and rotate while projecting the sternum anteriorly. As a result, the anterior, posterior and transverse diameters of the thoracic cavity increases. The increase of thoracopulmonary volume decreases pulmonary pressure to a volume of 756 to 757 mmHg, lower that air pressure by approximately 3 to 4 mmHg. As a consequence of these changes, a volume of air enters the lungs until the two pressures equalize. The volume of air that enters or leaves the lungs during resting or relaxed breathing is called current volume or tidal volume. So, students, now let us discuss expiration. Resting expiration is a passive phase. Expiration occurs when the thoracopulmonary structures return to the original position after the deforming force has terminated. As a result, the lungs decrease in volume and intrapulmonary pressure increases, making it higher than atmospheric pressure by 3 to 4 mmHg. This results in the elimination of a volume of air rich in carbon dioxide from the lungs into the atmosphere. Forced expiration is an active phase which takes place due to expiratory muscle contraction, muscles represented mainly by abdominal muscles and internal intercostal muscles. The contraction of the abdominal muscle increases intra-abdominal pressure, which in turn increases the convexity of the diaphragm and further reduces thoracic and pulmonary volume. Dear students, it's noteworthy to explain some more terms related to pressure changes in lungs, that are: pleural pressure. It's the pressure of the fluid in the thin space between the lung pleura and the chest wall pleura. The normal pleural pressure at the beginning of the inspiration is the amount of suction required to hold the lungs open to their resting level. Then, during normal inspiration, expansion of the chest cage pulls outward on the lungs with greater force and creates more negative pressure. The alveolar pressure. It's the pressure of the air inside the lung alveoli. When the glottis is open and no air is flowing into or out of the lungs, the pressures in all parts of the respiratory tree all the way to the alveoli are equal to atmospheric pressure, which is considered to be zero reference pressure in the airways, that is 0 centimeters water pressure. To cause inward flow of air into the alveoli during inspiration, the pressure in the alveoli must fall to a value slightly below atmospheric pressure, that is below zero. This slight negative pressure is enough to pull 0.5 liter of the air into the lungs in the 2 seconds required for normal quiet inspiration. During expiration, opposite pressure occurs. The alveolar pressure rises to about 1 centimeter of water, and this forces the 0.5 liter of inspired air out of the lungs during the 2 to 3 seconds of expiration. The transpulmonary pressure. The difference between the alveolar pressure and the pleural pressure is called the transpulmonary pressure. It is the pressure difference between that in the alveoli and that on the outer surface of the lungs, and it is a measure of the elastic forces in the lungs that tend to collapse the lungs at each instant of respiration, called the recoil pressure. Further, let us discuss the pulmonary volumes and capacities. A simple method for studying pulmonary ventilation is to record the volume movement of air into and out of the lungs, a process called spirometry. The tidal volume is the volume of the air inspired or expired with each normal breath. It amounts to about 500 milliliters in the adult male. The inspiratory reserve volume is the extra volume of air that can be inspired over and above the normal tidal volume when the person inspires with full force. It's usually equal to about 3000 milliliters. The expiratory reserve volume is the maximum extra volume of the air that can be expired by a forceful expiration after the end of a normal tidal expiration. This normally amounts to about 1100 milliliter. The residual volume is the volume of the air remaining in the lungs after the most forceful expiration. This volume averages about 1200 milliliters. And the pulmonary capacities are: the inspiratory capacity equals the tidal volume plus the inspiratory reserve volume. The functional residual capacity equals the expiratory reserve volume plus the residual volume. The vital capacity equals the inspiratory reserve volume plus the tidal volume plus the expiratory reserve volume. The third part of today's lecture, the alveolar ventilation. Alveolar ventilation is the exchange of gas between the alveoli and the external environment. It's the process by which oxygen is brought into the lungs from the atmosphere and by which the carbon dioxide carried into the lungs in the mixed venous blood is expelled from the body. Alveolar ventilation rate is the rate of the air flow that the gas exchange areas of the lung encounter during normal breathing. The alveolar ventilation rate is a critical physiological variable as it is an important factor in determining the concentrations of the oxygen and carbon dioxide in functioning alveoli. High rates of air exchange in functioning alveoli, that's higher alveolar ventilation, would bring in fresh oxygen-rich air and afflicts carbon dioxide-laden air rapidly. Consequently, the concentration of oxygen would be higher and the concentration of carbon dioxide would be lower within the alveoli. Alternatively, low rates of the air exchange in functioning alveoli, that is lower alveolar ventilation, would bring in fresh oxygen-rich air and afflicts carbon dioxide-laden air slowly. Consequently, the concentration of oxygen would be lower and the concentration of carbon dioxide would be higher within the alveolus. Now, measurement of the alveolar ventilation. The alveolar ventilation rates can be calculated using the alveolar ventilation equation described below. The alveolar ventilation equation takes advantage of the observation that all of the carbon dioxide exhaled by the body must come from gas exchanging areas of the lung, that is ventilated alveoli. It then normalizes this rate of carbon dioxide exhalation by the partial pressure of the carbon dioxide in the alveoli in order to correct for higher or lower values of carbon dioxide present in the alveoli. After discussing the alveolar ventilation, the next part is the gaseous exchange. So students, in this subtopic, let me first give you the definition of the respiratory unit. The respiratory unit is composed of a respiratory bronchiole, alveolar ducts, atria and the alveoli. The alveolar walls are extremely thin and between the alveoli is an almost solid network of interconnecting capillaries. Thus, it's obvious that the alveolar gases are in very close proximity to the blood of the pulmonary capillaries. Further, gas exchange between the alveolar air and the pulmonary blood occurs through the membranes of all the terminal portions of the lungs, not merely in the alveoli themselves. All these membranes are collectively known as the respiratory membrane, also called the pulmonary membrane. Here, it's important to give you a brief idea of the respiratory membrane. The respiratory membrane, also called the respiratory surface, has following characteristics. Number 1, it should be permeable to gases. Number 2, it should be thin to allow effective diffusion. It should be richly supplied with blood vessels or bodily fluids to allow maximum uptake of oxygen. It should have a large surface area to allow maximum uptake of oxygen in minimum time. Now, coming to the main topic, the exchange of gases. Dear students, the exchange of gases takes place at two sites, at alveolar surface and at the tissue level. Exchange along the alveolar surface. Each alveolus consists of a single layer of flattened squamous epithelium and is highly permeable and thin. The capillaries that form a network also have a layer of squamous epithelium. The barrier between the alveolar wall and the alveolar capillaries is extremely thin, and hence diffusion of gases takes place from higher partial pressure to the lower partial pressure. The blood that reaches the alveolus has lower partial pressure of oxygen and higher partial pressure of carbon dioxide than the alveolar pressure. As a result, the oxygen diffuses into the blood and carbon dioxide out of the blood into the alveolus. By the time blood leaves the alveolus, it has almost the same partial pressure of the oxygen and partial pressure of the carbon dioxide as the alveolar air. The percentage saturation of blood also rises from 70% to 95%. Now, exchange of gases in the tissues. In the tissues, exchange of the gases occurs between the blood and the tissue cells through the tissue fluids that surround the tissue cells. Blood that reaches the tissues has more partial pressure of oxygen than in the tissues. Similarly, the partial pressure of carbon dioxide is more in tissues than in blood. Due to these differences in the partial pressure of gases, oxygen from blood diffuses into the tissues and carbon dioxide from tissues diffuses into the blood. These exchanges of gases occur simultaneously. Now, moving to the last part of today's lecture, that is the transport of gases in blood. Owing to the different mechanisms of gaseous transport, we will discuss the transport of oxygen and carbon dioxide in blood separately. First, transport of oxygen in the blood. Although oxygen dissolves in blood, only a small amount of oxygen is transported this way. Only 1.5% of oxygen in the blood is dissolved directly into the blood itself. Most oxygen, 98.5%, is bound to a protein called hemoglobin and carried to the tissues. In solution, oxygen is soluble in plasma to a small extent under normal conditions of temperature and pressure. Hence, most of it is carried by RBCs. About 3% of oxygen is transported by blood in dissolved form in plasma of blood. As oxyhemoglobin, RBCs contain a protein called hemoglobin. It has 4 polypeptide chains and 4 heme groups attached to it, or 4 atoms of iron in ferrous form. Thus, it can react with 4 molecules of oxygen to form oxyhemoglobin, also designated as Hb4OH, in a combination process called oxygenation. Here, it's noteworthy to briefly discuss hemoglobin. Hemoglobin is a protein molecule found in RBCs made of 4 subunits, 2 alpha subunits and 2 beta subunits. Each subunit surrounds a central heme group that contains iron and binds 1 oxygen molecule, allowing each hemoglobin molecule to bind 4 oxygen molecules. Molecules with more oxygen bound to the heme groups are brighter red. As a result, oxygenated arterial blood, where the hemoglobin is carrying 4 oxygen molecules, is bright red, while venous blood, that is deoxygenated, is darker red. The binding of oxygen to hemoglobin can be plotted as a function of the partial pressure of oxygen in the blood versus the relative hemoglobin-oxygen saturation. The resulting graph, an oxygen dissociation curve, is a sigmoidal or S-shaped. As the partial pressure of the oxygen increases, the hemoglobin becomes increasingly saturated with oxygen. Oxygen dissociation curve. The oxygen dissociation curve demonstrates that as the partial pressure of the oxygen increases, more oxygen binds hemoglobin. However, the affinity of the hemoglobin for oxygen may shift to the left or the right depending on the environmental conditions. Now, moving on to the factors that affect oxygen binding. The oxygen carrying capacity of hemoglobin determines how much oxygen is carried in the blood. In addition, other environmental factors and diseases can also affect oxygen carrying capacity and delivery. The same is true for carbon dioxide levels, blood pH, and body temperature. Carbon dioxide level and pH. As the level of carbon dioxide in the blood increases, more H positive is produced and the pH decreases. The increase in carbon dioxide and subsequent decrease in the pH reduce the affinity of the hemoglobin for oxygen. The oxygen dissociates from the hemoglobin molecule, shifting the oxygen dissociation curve to the right. Bohr effect is an important phenomenon that shifts the oxygen hemoglobin dissociation curve to the right by increase in the carbon dioxide partial pressure. Body temperature. A similar shift in the curve also results from an increase in body temperature. Increase in temperature, such as from increased activity of the skeletal muscle, causes the affinity of the hemoglobin for oxygen to be reduced. 2,3-diphosphoglyceric acid. 2,3-diphosphoglyceric acid is present in the red cells of the adult blood formed in Embeden-Meyerhof pathway. It competes for oxygen binding sites in the hemoglobin molecule. As it binds to the beta chain of HbA, it causes a right shift of the dissociation curve. Diseases. Diseases such as sickle cell anemia and the thalassemia decrease the blood's ability to deliver oxygen to tissues and its oxygen carrying capacity. Now, transport of carbon dioxide in the blood. Several properties of carbon dioxide in the blood affect its transport. Carbon dioxide molecules are transported in the blood from body tissues to the lungs by 3 methods. Number 1, dissolution directly into the blood. Number 2, binding to hemoglobin. Number 3, carried as a bicarbonate ion. Dissolution directly into the blood. The carbon dioxide is more soluble in blood than oxygen. About 5 to 7% of all carbon dioxide is dissolved in the plasma and is carried in solution to the lungs. Binding to hemoglobin. Carbon dioxide can bind to plasma proteins or can enter red blood cells and bind to hemoglobin. This form transports about 10% of the carbon dioxide. When carbon dioxide binds to hemoglobin, a molecule called carb-aminohemoglobin is formed. When it reaches the lungs, the carbon dioxide can freely dissociate from the hemoglobin and can be expelled from the body as a bicarbonate ion. Third, the majority of the carbon dioxide molecules, that is the 85%, are carried as a part of the bicarbonate buffer system. In this system, carbon dioxide diffuses into the red blood cells. Carbonic anhydrase within the red blood cells quickly converts the carbon dioxide into the carbonic acid. Carbonic acid is an unstable intermediate molecule that immediately dissociates into the bicarbonate ions and hydrogen ions. Since carbon dioxide is quickly converted into bicarbonate ions, this reaction allows for the continued uptake of carbon dioxide into the blood down its concentration gradient. It also results in the production of hydrogen ions. If too much hydrogen ions is produced, it can alter blood pH. However, hemoglobin binds to the free hydrogen ions, limiting shift in pH. The newly synthesized bicarbonate ion is transported out of the red blood cells into the liquid component of the blood in exchange for a chloride ion. This is called the chloride shift or Hamburgers phenomena. When the blood reaches the lungs, the bicarbonate ion is transported back into the RBCs in exchange for the chloride ion. The hydrogen ion dissociates from the hemoglobin and binds to the bicarbonate ion. This produces the carbonic acid intermediate, which is converted back into the carbon dioxide through the enzymatic action of the carbonic anhydrase. The carbon dioxide produced is expelled through the lungs during exhalation. Dear students, that's all about today's program. Hope you have understood and enjoyed well. Thank you and goodbye.